Hello everyone. So now the third lymphocyte, that is natural killer cell. So we have already seen the details and features of B lymphocyte and T lymphocyte. They are performing a very significant role in immune responses. Alright. So here a little different lymphocyte we are going to discuss is natural killer cell or NK cell. So how they are different from other two lymphocyte is that this cell, NK cell, is basically associated with cell mediated mechanism, or a kind of innate response is provided by the NK cell. So they usually comprises around 5 to 10% of the total lymphocytes, and usually they are found in blood, lymph and other peripheral organs. Then similar to the lymphocyte like B cell, they undergo their processing like maturation and development in the bone marrow itself. Then after that, usually they move to the circulation, and from there they can migrate to the peripheral sites. Then they are having a kind of immediate response capacity, and hence they are part of the body's defense in a very significant way. They act as the first line of defense against, mostly against viral infections and also against tumor development. They can do the self renewal, and usually they persist in the host or in the body for months to years. That means they have a comparatively longer lifespan. They survive for a long period of time in the body after synthesis and release into the circulation. Then these are sometimes also called as large granular lymphocytes or LGLs. As it suggests, there are granules in the cytoplasm, and these granules are usually filled with chemicals which are toxic to most of the target cells, like these chemicals collectively called as perforins and granzymes. So these cells were sometimes also referred as null cells, because these are non-B, non-T cells. That's what NUN indicates. These are not similar to B cell or not similar to T cells. That's why they are referred sometimes as null cells. That means previously they were considered as null cells. Basically, why they are called null? Because there is no specific receptor within this particular group of cells. There is no specific receptor similar to that of a B cell receptor or a T cell receptor. Right. So you can conclude that there is no specific unique receptor for NK cell, just like T cell receptor for T cell and B cell receptor for B cell. There is no such receptor for NK cell. Right. So, but there will be some common receptors as shown here. There will be receptor like CD56, CD16 or the Fc gamma R3 receptor, then some other CD2 receptors and CD8 receptor. All those are common receptors found in lymphocyte, but there is no unique receptor for any NK cell category. Then one of the most important question is that how this NK cell identify the target cell. In the previous videos, we have seen that how B cell recognize antigens, and also how T cells recognize antigens. We know that B cells will interact with the foreign substances through the interaction of B cell receptor. Similarly, T cell will interact with antigens through the T cell receptor, and the antigen, already we have discussed in detail that the antigen to be supplied along with a self molecule called MHC, and usually done presented by the antigen presenting cells or altered self cells. Right. So here the point is that there is no such receptors. There is no B cell receptor and no T cell receptor. So the question becomes relevant. That means how these NK cells will identify the target cell. That is an important question to be addressed. Right. So here there are two different ways how these NK cells will identify the target cell. Right. So before that, you have to understand that what are the target cells. The target cells are basically normally these are the viral infected cells or the tumor developing cells. Right. So how they identify? The first method or the first way how this NK cell will identify a target cell is that they will check for a reduction in class 1 MHC receptors on the surface of a normal cell. And we know already that for every nucleated cell, the class 1 MHC is a common receptor. right? There will be a class 1 MHC molecule for every nucleated cell, which means that all cells of the body consist of class 1 MHC, and there will be a particular concentration, there will be a particular number of class 1 MHC molecules usually expressed on the surface when the cell is in a normal situation, right? And there will be a change, there will be a reduction expected when there is a damage, when there is a viral infection, or when there is a kind of tumor development by the cell. So in that case, NK cell will screen the cell for the concentration of the MHC class 1 molecules on the surface, and will recognize these cells having less expression of class 1 MHC. And this will indicate that there is some problem inside this self molecule, in cell, so that NK cell will identify this molecule as a target, and that will be destroyed. So that is what's happening, right? And the detailed mechanism we will see in the next part, next slide. And the second part, or the second method, second way of recognizing a target cell is by a mechanism called ADCC, or antibody dependent cell mediated cytotoxicity. Antibody dependent cell mediated cytotoxicity. That is a very important mechanism, usually coming in the innate mechanisms. Right, here there are we know that on the surface of NK cell there are receptor like CD16, right? There are receptor like CD16, or this is otherwise the FC gamma R3 receptor. So this receptor is a complementary binding site for the FC portion of antibody molecules, and those antibody molecules which are recognized, those antibody molecules which are bound on certain antigens, that means here antigens are nothing but the antigens expressed by the target cells. So such cells will be bound over by the antibody molecules, and these antibodies will interact with the NK cell. So this interaction also will activate the NK cell to secrete chemicals, right? So these kind of cells with CD16 receptor and having the capacity to perform ADCC are sometimes referred as killer cells, right? So these are also sometimes called as killer cells if they are performing antibody dependent cell mediated cytotoxicity. Okay, then so the same story is explained here. So while in the circulation, for the first mechanism, while in the circulation it will identify and bind to the class 1 MHC molecule on the surface of all nucleated cell. This is what the first point. Then attack by an intracellular pathogen like virus or a cancer development will cause a reduction in the class 1 MHC, right? Then this reduction in MHC concentration on the surface of the cell will activate the NK cells, and that leads to the target set, right? Then automatically there will be secretion of chemicals to destroy this targeted cell. So here we will explain a little more about this, how this MHC signal is being associated with the recognition, right? So look at this image, you can see there are two signals. The first signal shown in green color, the NK cell has a receptor called activating receptor, and there is a complementary ligand on the target cell, that means on the self cells, okay, self host body cells. So this interaction leads to an activating signal, right? That is a positive signal that the NK cell will be activated, and this activation of NK cell will lead to the secretion of chemicals and destruction of the target cell. This is a persisting signal. This signal is there for every NK cell self-cell interaction, right? But the effect of this interaction, effect of this positive signal is nullified by the presence of class 1 MHC molecule on the surface of target cell. If there is enough quantity, enough concentration of MHC class 1 molecule on the target cell, they interact with the complementary receptor on the NK cell, and this interaction will create an inhibitory signal, and this inhibitory signal will nullify the effect of this activating signal. That is the brief story. So let's see the discussion. So that's what I already told you. that there are two types of receptors or two types of signals generated. One is the activation signal or activation receptor signal. Then second one is an inhibitory signal, and these inhibitory signals are crucial because they have to counteract the killing effect of the NK cells, which is being responsible, which is being credited by this interaction of the activating receptor and its corresponding ligand. Right. So, the AR receptor on NK cell and its ligand on the cell gives a killing signal, whereas the MHC1 and its ligand gives the inhibitory counter effect. Fine. So, so now you can understand how this MHC reduction will lead to a destruction of the target cell. Right, because if the MHC class 1 is not expressing normally, if there is a reduction in the concentration, then only the positive signal persists. Right, only this signal will be the predominant signal available, so that and there is no inhibitory signal, so the killing will be the result. All right, so hope you understood this mechanism. So this image will give you a little more idea. So here you can see this is the negative signal and this is the positive signal, right? And the positive signal is actually nullified by the negatives. If there is a negative signal, then the positive signal will not be affected, so that there is no killing. All right, but when there is no negative signal, because there is no MHC expressed here, so if there is no MHC molecules found on the surface of the target cell, then automatically NK cell will identify this as a target because only the positive signal persists. Then there is a destruction. Right. So this is about the mechanism by which this NK cell will identify the target cell by means of reduction in class 1 MHC. That is the first mechanism. All right, there is a reduction in MHC class 1 if the target cell has viral infection or which is a tumor developing cell. In that situation, the MHC will be reduced and the NK cell will target this as a target for destruction. Fine. Now the second mechanism, ADCC. So this image will give you a detailed idea about ADCC. So if a cell infected by a virus or an intracellular parasite may express certain antigens on their surface, you can see there are certain antigens which are expressed on the surface of this is the target cell, right, which is having an infection. Actually it is a viral infected cell, and there are certain antigens expressed on the surface, right, and these automatically what happens, the immune system or immune cells will identify the antigen and the B cells will produce antibodies against the antigen. That already we know that mechanism. Now this antibody will bind to the antigen on the surface of the cell with the infection, right. So this target cell with the antigen will be bound by these antibodies. So these antibodies are produced by the B cells against these antigens. Fine. Now the NK cell will interact with the FC portion of the antibody. We know that this is the FC portion. These are the FC portions of the antibody molecules, and this NK cell has certain receptors, what we call the CD16 or the FC gamma R3 receptors. So these receptors are complementary to this FC portion of the antibody molecule. Right. So what happens ultimately? What happens? This FC portion will be bound over to this FC gamma R3 receptor, so that there is a linking between this NK cell and the target cell. So this interaction is made through the CD16 receptor on the NK cell, and which ultimately leads to the degranulation and release of the cytotoxic chemicals and destruction of this target cell. All right. So this is the second mechanism. This is what we call the ADCC or antibody dependent cell mediated cytotoxicity. So these kind of cells are sometimes referred as killer cells, as already we mentioned. So that's about the mechanism of NK cell destruction. Now a variant of NK cell, we call it as NK T cell, right? NK T cell. So why it is called? You can understand that why it is called NK T cell, because they are showing characteristics of both T cell and NK cell. That's why it is called NK T cells, right? So what are the features related to T cells? The first feature is that they have a T cell receptor, right? They show a T cell receptor, right? But the T cell receptor is not similar to that of the T cell receptor on the T cells, but still there is a T cell receptor, but this TCR will not interact with MHC molecule, but instead they interact with another receptor called CD1, right, CD1 on the target cell, right, and moreover this TCR is not that much diverse, right, it is not diverse for interaction with antigens. And similarly, they are matured in the thymus. The maturation process of this NK T cell occurs in the thymus. So these two are the features related to or similar to that of T cell. All right, but similar to or how this is related to NK cell is that they shows receptors like CD16, right. The CD16 is a receptor commonly found on NK cells. So they are having this CD16 receptor, and moreover they have this capacity to secrete chemicals, and these chemicals are also similar to that of NK cells. So these cells shows properties of both NK cells and T cells. That's why they are called as NK T cells, right. But the concentration is comparatively very less, 0.01 to 1% of the peripheral blood mononuclear cells. That means only 0.01 to 1% of the out of this total cells, only 0.01 to 1% will be this NK T cell, very rare, right. And they are showing some kind of immunoregulatory properties also. Okay. They secrete chemicals like interferon gamma, and these chemicals will have some significant role. So that's about these NK cells. So NK cell, I'll brief once again. NK cells are the lymphocytes without a specific unique receptor. These are otherwise called large granular lymphocytes. The cytoplasm is rich with granules, and these granules are filled with cytotoxic chemicals, right. They are associated with cell mediated immunity or innate mechanism, right. They have certain other receptors like CD56 and CD16, fine. Then the way how they interact with the target without a receptor is by two mechanisms. One is by screening for the class 1 MHC. If there is any reduction in class 1 MHC, that particular cell will be targeted. The second mechanism by ADCC, antibody dependent cell mediated cytotoxicity, and by that also the cell will be targeted. And finally, we have seen a sub variant of this NK cell called NK T cell, which shows properties of both NK cells and T cells. So that's about NK cells. Hope you understood. Thank you.